It's now a distinct pleasure to invite our second plenary speaker for this session, and that is Stephen McCarroll, who came here from Harvard Medical School in Boston in the US. And Steve is a fantastic scientist and pioneer in many aspects, I have to say, and we've been very closely following his work for years, and I think he's not always visible on the ECG stage, but a lot of his work is really truly stunning. And several of his discoveries and also pioneered technologies all came together for a really fantastic discovery from last year. And we're so excited to have you here to speak about some of the underlying fundamental issues in Huntington's disease, which may well translate to other repeat expansion disorders. So it's a true privilege to have you on stage, Steve. The floor is yours.
Thank you so much for inviting me to come and share this work. Human genomes have tens of thousands of loci at which short DNA sequences, such as CAG, are repeated a substantial and variable number of times in different people. More than 50 human genetic diseases are known to be caused by inherited alleles in which one of these repeats has expanded beyond its normal range in human populations. Most of these were discovered in the last few years with long-read sequencing, and very many are diseases of the brain. The classic DNA repeat disorder is Huntington's disease, a fatal genetic brain disease. People who eventually develop Huntington's first have decades of apparently healthy life with no signs of the calamity that's going to befall them later. Symptoms typically begin in midlife. The most apparent symptoms are that it's a movement disorder with progressive writhing, involuntary movements that become worse and worse, the decline, psychiatric symptoms, and then the movement disorder progresses over 15 years to debilitation and death. There's no effective treatment. It's always fatal. In Europe and the United States, where Huntington's disease is most common, there are about 60,000 patients and another 400,000 people who are at genetic risk as first-degree relatives of people with the disease. Huntington's disease involves profound atrophy of the striatum, shown here in the brain imaging. And within the striatum loses its primary neuronal population. It's not its only neuronal population, but it's its largest one. These are called striatal projection neurons, or SPNs, also historically known as medium spiny neurons, or MSNs. And science has long struggled to understand why this particular kind of neuron is so vulnerable when the cells around it all survive. Huntington's disease is the textbook Mendelian human genetic disease. In fact, many of us will have learned Mendelian disease genetics in humans through the prism of Huntington's disease. It has autosomal dominant inheritance, full penetrance, very large families with many affected family members across many generations. And these families were used to, this was actually the first autosomal dominant disease gene to be molecularly mapped to a human chromosome in 1983, and then identified by gene mapping and positional cloning in 1993. The consortium that discovered it named the gene Huntington, or HTT. And this was such a watershed discovery for human genetics that its success in many ways spurred the Human Genome Project to create a knowledge base with which to discover many more such things more quickly, as in fact has happened. Huntington's disease is caused by an inherited DNA repeat, a CAG repeat, in the first exon of the Huntington gene. Most of us inherit from our parents alleles with 10 to 30 CAGs in the first exon. Everyone with Huntington's disease has inherited an allele with 36 or more CAGs, typically 50 or more. And as you see in this plot on the right, there's this relationship between the length of the CAG repeat that one inherits and the age at onset, where across this range of 35 to 50 CAG repeats, longer inherited alleles correlate with earlier onset of symptoms. And the prevailing biological interpretation of this for the past 30 years has been that these inherited alleles encode a polyglutamine stretch that is toxic, but only slowly toxic, requiring many decades to accumulate in a way that brings about the decline and death of striatal projection neurons. But the fundamental biological mysteries about Huntington's disease have endured for the past 30 years. What is neurotoxic about the inherited alleles that cause this disease? Why do only some kinds of neurons seem to be sensitive to this toxicity when all cells express the Huntington gene and protein? And why is there this decades-long latent period in which individuals seem completely healthy with no sign of the calamity that will befall them later? 10 years ago this week, our lab described what we called DROP-seq, or droplet-based single-cell RNA-seq, a new technology for using tiny aqueous droplets created by microfluidics as tiny reaction chambers in which to do high-volume single-cell RNA-seq. Naturally, and this of course has evolved into things like the 10x genomics technology. Naturally, this seemed like a good approach to apply to a problem with such cell type specific pathology. And we started by doing the obvious thing. We analyzed the striatum, in particular the caudate nucleus, from 50 people with Huntington's disease and 50 controls. We did the standard analyses, assigning cells to cell types based on their gene expression patterns. And one thing that we see that's not surprising, but is still useful to quantify, is the really striking loss of striatal projection neurons as the disease progresses. Here we have the 50 controls on the left, and on the right, the 50 cases ordered by increasing CAP score, where CAP score is a prognostic indicator calculated from age and inherited CAG repeat length to kind of forecast clinical trajectories. And what you can see is the profound loss of SPNs, the dark orange bar, as the disease progresses. This is the same data on a log scale and with SPN survival on the y-axis. And you can see that for a very long time, there's essentially no loss of SPNs. Then SPNs begin to be lost, and then they're lost at escalating rates until the end of life. Of course, another thing one can do is compare gene expression in each cell type between cases and controls. And here we reach the kind of intellectual dead end that descriptive functional genomics so often arrives at, which is that we find that every cell type has thousands of genes that differ in expression levels on average between cases and controls. And in some ways, this is not surprising. This disease causes profound atrophy of the brain, loss of neurons, devascularization. And of course, this just profoundly changes the biology of every cell type in the brain. But this doesn't help us understand what is the first order thing that the CAG repeat is doing to the biology of cells. This is just downstream sequelae. That is to say, what are the causes and the effects? So it was what we did next that really changed our understanding of this. And before I tell you about this, I just want to say, this was, unlike DROP-seq, this was not just an idea we had and drew up on a whiteboard in the lab one day. This really, our focus on somatic expansion really came from earlier work in the field that I want to describe to you on the next few slides. So the CAG repeat that causes Huntington's has been known for 30 years to be somatically unstable. This is work from Peggy Shelburne's lab and others showing that the DNA molecules sampled from the brain contain a variety of CAG repeat lengths. And work from Vanessa Wheeler and Ricardo Mauro Pinto and others has shown that this somatic expansion of this repeat is actually caused by our cell's own DNA maintenance proteins through a DNA repair process that I'll tell you more about in a few minutes. But it was a recent human genetics discovery that really drew our attention to somatic expansion as a key intermediate phenotype to try to measure at the single cell level. So although whether you get Huntington's or not is Mendelian, age at onset is a quantitative trait. It's only partially predicted by the length of the allele that one inherits. There's still a lot of variance across individual patients. And this difference, the residual, this difference between actual and predicted age at onset was used as a quantitative trait for a genome-wide association study that found two kinds of surprising things by the GEMI-HD consortium. First, the CAG repeat contains synonymous substitutions that don't change the encoded protein, but profoundly affect age at onset. And this tells us that something important must be happening at the nucleic acid level. Secondly, more than half of the loci at which they mapped common modifiers at age of onset contain DNA repair proteins. And not just any DNA repair proteins, these are precisely the genes that have been shown to affect the stability of DNA repeats in the work I told you about a couple minutes ago. So we spent quite some time developing an approach for single-cell analysis of the HTT-CAG repeat alongside the genome-wide RNA expression of each cell. So in addition to the kind of standard DROP-seq or 10x genomics workflow, we also prepare from the same set of nuclei and barcoded nucleic acids another library that contains each cell's CAG repeat sequence. And the presence of a set of shared cell barcodes that we apply at this stage allows us to connect each CAG repeat sequence to the genome-wide RNA expression and thus the cell type and biological state of the cell from which we sampled it. So what cell types actually harbor this somatic CAG repeat length mosaicism? We applied our approach to postmortem tissue from people who had Huntington's disease during their lifetimes, and this result was really clear. The CAG repeat shows profound somatic expansion in SPNs, the cell type that dies in Huntington's disease, and not in the other cell types of the striatum. And this expansion is also allele specific. It's exhibited by the allele that causes Huntington's disease, but not by the normal, common, shorter allele that's present in the same patient. So it's both allele-specific and cell-type-specific expansion. And this is true in every patient that we've looked at. So the mysterious cell-type specificity of the pathology in the illness corresponds to the cell-type specificity of somatic expansion of the DNA repeat. You might wonder, how is this even happening in neurons? Neurons are non-dividing cells. They're post-mitotic. In this slide, I just want to tell you what work from other labs has taught us about the mechanism of DNA repeat expansion in non-mitotic cells. So whenever the two strands of DNA come apart, for example, for transcription, there's a chance that they will come back together in an out-of-register manner, like a zipper with a pucker. And when this happens, this gives rise to these slip-out structures. And these slip-out structures get surveilled by DNA maintenance complexes that are constantly combing through our genomic DNA looking for mismatches. And this complex mut-S initiates a repair pathway with mut-L causing a nick nearby. And this strand is then excised and resynthesized using the other strand as a template. And you can see that what this results in is, if these two slip-out structures are far enough apart, more so than the excision distance, is this results in a length change mutation. So we noticed that patient after patient has what we call an armadillo distribution of CAG repeat lengths, in which typically 90 to 98% of their neurons at the end of the neurons they still have at the end of their life have expanded from 40 to about 80 CAGs. But there's always this long tail of a few percent of neurons with much longer expansion. So how do these arise? We approach this question through mathematical simulation, mathematical models and computational simulations. So here you can see one of these simulations. We just make simple assumptions. Okay, first that these expansion events are stochastic events at the single cell level. And secondly, that it's nonlinear. The longer it repeats, the more frequently these events start occurring. And these models end up generating end-of-life distributions that strongly resemble the actual end-of-life distributions that we're measuring through single-cell analysis. And in particular, our model predicts that there's a transition from slow expansion to a much faster rate of expansion at about 80 CAGs. So this somatic expansion seems to proceed in two phases that we call phase A and phase B. And note that although phase B involves expansion of many more CAGs, neurons spend much more of their lives in the slow phase of expansion, which we call phase A. And then they enter phase B asynchronously at different times because this is a stochastic process. But the key question for the past 30 years has been how and when does the CAG repeat change the biology of the neuron that it's in? As I mentioned, if you just compare gene expression between cases and controls, We see thousands of changes in every cell type, but this doesn't help us understand what's the direct cell-autonomous consequence of the CAG repeat. But we had an idea. We realized that within every little piece of brain tissue, there's a whole allelic series of thousands of individual neurons, each with its own CAG repeat length, because it's a stochastic process. And if we compare these neurons to one another, it allows us to isolate the direct effect of this CAG repeat length on the individual neuron that it's in. So what happens if we compare the length of this, if we compare neurons with some expansion, let's say 35 to 65 CAGs, to neurons in the same patient with 66 to 150 CAGs? What you can see from this volcano plot is that we see no gene expression differences. That is to say, it's as if the CAG repeat has no effect on a neuron's biology within this range. But what if we compare these neurons to neurons way out on this long tail with much longer CAG repeats? Then we see thousands of gene expression differences. This is now data from 6 patients. In each patient, we've made a genes by cells, a heat map, where color represents fold change relative to baseline. And we've just ordered the individual neurons from each patient from left to right based on the length of their CAG repeat. And what you can see, just visually even, is that there's profound gene expression change, but just in the neurons that have expanded beyond 150 CAGs. Of course, this data is noisy at the single cell, single gene level, but we can collapse the measurements from many genes in the same cell into a metagene and ask what's the median fold change of those genes. And we make 2 metagenes, one for the genes that go up and one for the genes that go down. And what you can see is that for both genes, there's essentially no change in their expression levels until the repeats expanded beyond about 150 CAGs. And then there's profound and escalating change. So we can now add a toxicity phase, a phase C of steadily escalating transcriptionopathy that we think, based on these simulations, lasts just a few months at the end of a neuron's life. Then there's a final phase. So in neurons with the most advanced phase C transcriptionopathy, and also with still longer CAG repeats, we see de-repression, unsilencing of more than 100 genes that are normally silent in adult neurons. These include the genes in the Hox clusters, but also genes like CDKN2A and CDKN2B, which promote senescence and apoptosis. And in fact, expression of CDKN2A, when forced in adult neurons, has been shown to be toxic to neurons. And so we think this is what ultimately brings about the death of these neurons. So our expectation at the time we started this work was that with this approach, we could characterize the direct biological effects of an increasingly toxic mutant Huntington. But these experiments led us to a really different way of thinking about this disease. What we now think is that for more than 95% of a neuron's life, the pathological process is really just more like a slowly and silently ticking DNA clock, in which the CAG repeat is expanding a little bit, initially less than once a year, and then more and more quickly, stochastically and asynchronously across the individual neurons. But as the repeat gets longer, it starts to expand more quickly, and so this DNA clock starts ticking much more rapidly and predictably, but still without an immediate biological consequence. Only once the repeat expands beyond what we now believe is a very high toxicity threshold does it even begin to have biological effects that we can detect at the level of a neuron's own gene expression. And these effects escalate until they ultimately bring about the demise of that cell a few months later. So let's return to these biological mysteries about neurodegeneration and Huntington's disease that have been so challenging to answer. What is neurotoxic about the inherited alleles that cause HD? Well, we think that the reason this question has been so hard to answer for the past 30 years is that nothing's toxic about them. We think that the inherited alleles are actually innocuous. Why does neurodegeneration take decades? Another really hard question to answer. We think the answer is that it doesn't take decades, that somatic expansion to this high threshold takes decades, but that neurodegeneration, once it starts, is fast, is months, not decades. And why is this neurodegeneration so cell type specific? We think that it's because somatic instability rates vary greatly across neuronal types. So we now think, based on these experiments, that the pathogenesis of Huntington's disease is a DNA process rather than a cell biological toxicity process for 95 to 99% of a neuron's life. So you may be familiar with these, the many efforts in advanced clinical development to treat Huntington's disease by lowering expression of what the conventional biological model believes to be a toxic Huntington protein. And there's a variety of approaches in advanced clinical development, ASOs, splicing inhibitors, other approaches. You may also be familiar with the surprising and very disappointing failure of these approaches to establish efficacy on clinical outcomes in clinical trials, even though they're very effective at reducing expression of Huntington in both preclinical models and human patients. So we think that these results give us some insight about at least one reason why Huntington-lowering therapies have been struggling to establish efficacy. The longstanding biological model is one of long and synchronous and indolent toxicity in which all neurons are slowly getting worse throughout a person's life under the influence of a toxic protein. But what we think this data suggests is that the toxicity in neurons is actually short, asynchronous, and intense. And that at any one time, only a very small fraction of neurons have a toxic protein entity whose expression one would even want to lower. But I want to leave you with a hopeful thought. It might be possible to instead slow the ticking DNA clock and to address this pathological trajectory back here at the stage at which most neurons spend 95% of their lives. Because there's so many more neurons without a toxic Huntington than with it at any moment in time, we think that this is potentially a far larger therapeutic window, both in terms of number of cells and amount in time. And as I mentioned before, our own enzymes are complicit in somatic expansion, and it may therefore be possible to slow somatic expansion by slowing the function of certain enzymes. An important insight comes from human genetics, such as the modifier analysis that I told you before. So, for example, there are two common haplotypes of the MSH3 locus that independently both delay the onset of Huntington's disease. By combining that genetic data with the genotypes from our donors and our single-cell data, what we can see is each of these haplotypes, for each of these haplotypes that postpones Huntington's onset also reduces the expression of MSH3. So reduced function is actually onset postponing. But these haplotypes only reduce MSH3 expression by about 7%. That's what my doctor calls a homeopathic dose of MSH3 lowering. And of course, any pharmaceutical effort would try to reduce function by much more. But human genetics also offers a hint that much deeper reductions may be safe. For example, data from Nomad indicates that there are very many people walking around already with heterozygous loss of function alleles of MSH3. In fact, it doesn't seem to be under purifying selection. And analysis of those UK Biobank participants with these loss of function alleles suggests that there's no apparent evidence that they're at increased risk of cancer. So we think this is a promising therapeutic strategy that really deserves, and this idea was came out of the GWAS, but we think this really deserves intense focus because it there's potentially a lot of therapeutic leverage on the ticking DNA clock. Finally, we think that what this has taught us will probably apply to many DNA repeat disorders, not all, but many. And I want to point you to a preprint of some other work we've been doing together with Poru's lab. This is actually led by Margot Hujol, in which we find that DNA repeats at many genes actually expand throughout our lives. You can even see this in the blood from UK Biobank participants. And that these include somatic expansions in genes for many other DNA repeat disorders, and that these expansion rates are influenced by many of the same modifier genes and alleles that affect age of onset in Huntington's disease. So it may be, we hope that if down the road it becomes possible to address this mechanism in Huntington's disease, that such therapies may offer benefit in many different DNA repeat disorders. So to summarize, the alleles that cause Huntington's disease we think are initially innocuous, and they're not born bad. They must greatly expand before becoming toxic. That the pathogenesis of this disease is a DNA repeat expansion process for almost all of a neuron's life, and that the cell type specific vulnerability in Huntington's disease arise from cell type specific rates of somatic expansion in the DNA repeat. So some of the things that our lab is working on now, we're trying to understand what is the cellular toxicity mechanism that actually commences as the repeat expands past this 150 CAG threshold. We're trying to understand why are only some cell types vulnerable to somatic expansion, or why are some more vulnerable than others? How are these genetic modifiers acting upon this neuropathological trajectory? And finally, can this model of acquired toxicity, in which the alleles that cause disease are born innocuous but become toxic through somatic expansion during our lifetimes, can it help us understand some or even many of the other DNA repeat disorders in humans? So as you guys know, the people are the best thing about this kind of work, and I want to thank the members of this team that really brought together people with backgrounds in neuroscience and statistics and human genetics, and in particular the lead three scientists on the study, Bob Hansiker, Seva Cashin, and Nora Reed, and Sabina Beretta, who's been my partner in planning and thinking about this project since we first conceived it. And this team has taught me a lot and taught one another a lot. And I want to thank you for inviting me, and I look forward to your questions and thoughts and ideas. Thank you very much.
Steve, that was wonderful. Thank you so much. I think we've got time for a couple of questions. I see one right in the middle at the back, please.
Sure. So Winston Smith, Johns Hopkins University. Great talk. My question is, you're dividing into cases and controls, right? But you also told us that where you start matters for age of onset. What about if I had like 34 repeats? Is it just that we don't live long enough for that to start to have an impact because of this slow initial phase that's decades? Or have you looked at anybody who happened to have 34 repeats and what happens with them?
Yeah, so for a long time, 36 CAGs is thought to be the threshold for toxicity. But what we think is that it's actually the threshold for instability. And that the level of instability is actually highly nonlinear in this range of 36 to 40, and that not increased toxicity, but that is the reason that the age of onset is so much later if you're born with a 36 than if you're born with a 40. So then the question becomes, well, why is that the threshold for somatic expansion? We don't know the answer to that question, but we think that it relates to the excision distance of mut-L. So if you remember that slide where it cuts at some distance from the slip-out structure, if it cuts all the way over on the other side of the other slip-out structure, then you don't get expansion or contraction. So we think that ends up giving rise to a threshold below which there's no instability at all, at least in this cell type.
Very cool. Thank you.
Another question over on the left. Thank you.
Thank you very much for this really brilliant talk from Hildebert from Cologne. So my question is, since you have obviously answered to so many brilliant questions already, do you know why most of the time HD is paternally inherited when not paternally, but the repeat expansion is of paternal origin? While, for example, myotonic dystrophy is maternally? Is there any?
Yeah. So the disease-causing allele can be inherited from either parent, but it does seem to be unstable in the paternal germline, the male germline, and not the female germline, and we don't know why.
Okay, that was the question. Why?
That's fantastic. Thank you so much, Steve. Steve will be around to answer other questions.